Welcome back. Today's notes are on translation, which is the second and final stage of protein synthesis. Translation is the process of creating a protein, and this is done from the information that's in the mRNA molecule. We learned that during transcription, mRNA is used to code the instructions in DNA and take it out into the cell. So just a quick review, replication is making a copy of DNA, so DNA to DNA, and this happens in the nucleus. Transcription is converting DNA into mRNA, which also happens in the nucleus because we need a DNA template. And translation is converting mRNA into protein, and this will happen at the ribosome. I think a good way to, if you get the processes confused, for transcription, I think of a scribe, which is someone who would copy something down. In translation, we are converting the language of mRNA into protein. A codon is 3 nitrogen bases in an mRNA molecule. And remember, M stands for messenger, so it carries the instructions or the message for how to make a protein from DNA. Each codon is going to provide or code for a specific amino acid, and it's the instructions for getting that specific amino acid into the protein chain. So if you look at this sequence on the right here, that would be an example of an mRNA sequence. mRNA always starts with this AUG, which codes for the amino acid methionine. You don't need to know that, just fun information. And you'll see it's broken into groups of 3 different bases, and each one of those is a codon. And we know that based on the sequence of bases, we get different characteristics, and those are expressed through the different types of protein. There are 20 amino acids total, but you'll see that there are many different codons. So this is called a codon chart. There are 2 different formats of it, and you'll see the second one soon enough. This is used to tell us what amino acid a codon is coding for. So if you look down here, the codon CGG, we always start here on the left side with this format of the table for our first letter. Across the top, we look for the second letter. And then on the right, we have these groups of 4 for the 4 bases in RNA for the third letter. So basically, you're looking for the point that all 3 of these sections intersect. So again, if we look at CGG, I'm going to start with my first letter, C, and then G is going to take us to this far column on the right. And then my third letter, G, shows us that it is arginine. And you'll see there are many different codons that possibly code for one specific amino acid. Okay, let's look at another. GAA would be glutamate. So again, we start here on the left side, G's on the bottom. And then we go to A, which is the third column. And then I find my third A on the side here. And that gives us glutamate. So again, you're looking for the intersection of those 3 letters. Okay, GGG, I skipped ahead a little, but see, G gives us glycine. And then UGA would code for stop. And what stop means is that there is no amino acid there, and that is going to signal the ribosome to release the protein chain so that it can fold and go carry out whatever function it is supposed to do in the cell. There are 3 different stop codons, UAA, UAG, and UGA. So now it's your turn to try. What would GCC code for? Just give you a second to figure it out. Going to be alanine. So you see here, G, C, C is alanine, and AGC would be serine. Alright, this is the other form of the codon chart. It's much more recent. It came out in the past maybe 5 years or so. And the way this one works is you always start from the center and you work your way out. So it's a little more simple in my opinion, so less confusing. So if we were going to find GUC, I would start in the center, G, and then I go to the second ring, the middle ring, for U, and then I find C, and you'll see that that is valine. So it's a little less redundant, like it's not repeating the amino acids in different places all the time. So what would CAC be? If you got histidine, then you are correct. So again, we start in the center, C, A, and then I go to C here, and that would be histidine. There are versions of the chart of both kinds that will only show abbreviations. You can always just write the first 3 letters of the amino acid. Alright, and then lastly, UGA would code for stop. Okay, so again, that's another sequence or another codon that would signal the ribosome that that protein chain has ended. Alright, so in this case we are bringing all 3 types of RNA together. We talked about mRNA, which is our messenger, which carries the instructions to build the protein from the DNA. And then we have a tRNA molecule, and T stands for transfer. So this specific type of RNA is going to carry amino acids to the ribosome so that it can build a protein chain. Every 3 bases in a tRNA, there's a specific section on the bottom of that molecule, and you'll see it's kind of shaped like a T, and that is called an anticodon. So anticodon binds to the codon. This anticodon matches with the codon to ensure the correct amino acid is placed into the protein. So it will go onto the mRNA sequence, and if it base pairs with that codon, that amino acid is transferred to the chain. So that is how we determine the sequence of the proteins. So if our mRNA codon was AUG, which is what we always start with, our tRNA codon would be UAC, and then that would transfer methionine. So for our steps or process of translation, step 1, mRNA molecule arrives at the ribosome. It's going to attach to the ribosome. You may also see the term rRNA. rRNA makes up the ribosome. So it is ribosomal RNA, and the RNA combined with proteins makes up the structure. So step 1, mRNA molecule arrives at the ribosome. Step 2, the codon waits for the corresponding tRNA anticodon to arrive. So again, we're looking for that accurate base pairing between a certain tRNA and the codon so that that amino acid that's correct can be transferred to the chain. Step 3, attached to the tRNA is the correct amino acid. Once they base pair, we know that for sure. Step 4, the ribosome is going to shift. It's going to move over to the next codon so that now we have an available space for the next tRNA to come in. And then the amino acids will join into a chain and eventually form a protein. So you might see the term polypeptide, and that is the chain of amino acids. Proteins cannot function unless they are folded into their 3-dimensional shape, which hopefully you remember from our unit on enzymes. Here's a different picture showing a little bit more of these parts interacting. So again, we have our mRNA strand. It's a single strand. The ribosome is moving along the mRNA, and those tRNAs are going to come in sequence once they have found the right open space in the ribosome. So as the ribosome shifts, there'll be like an opening, and the tRNA will come in. Once the right one base pairs, then it'll add its amino acid to the chain, and then the empty tRNA that no longer has an amino acid will be released, and eventually it will get recycled so that that specific amino acid it carries, a new one will be added and it can go back to building proteins. And as I mentioned before, you see in this caption, there are 20 different amino acids, but depending on their sequence and how many there are, how many chains are involved in the folding, that will determine their function. There are a lot of moving parts in this process, so I'm going to show you a video as usual. Hopefully it will make it a little bit clearer for you.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Alright, for a quick recap, so the mRNA binds to the ribosome. Our tRNA is going to bind, so anticodon on the tRNA binds to the codon that is properly base paired on the mRNA. That amino acid is transferred to the growing chain. The ribosome moves along the mRNA, and that process is repeated where the proper tRNA comes in, transfers its amino acid over, and then eventually we reach a stop codon where the polypeptide or the amino acid chain is released, and it will fold into its proper shape so that it can do its job. So make sure that you write your summary in at least one question, and then come check in with me so I can give you a stamp.